Hi again. We have just talked about gene expression. This process how the information in DNA of the nucleus is made into something useful at the ribosomes. But not all genes are expressed at all times, otherwise this would be a chaos. After all, using the analog that I made earlier on, you do not want to cook all the recipes that you know at all times when hungry. Instead, you choose what is useful of those at that time. And I am going to do a bit of a deviation from your textbook here and leave a discussion of prokaryotic gene expression a bit later and instead discuss that in eukaryotes first. And actually, we are going to keep this very brief as I want you to study it in more depth from your textbook. But let's make a start nonetheless. So, as I said, not all genes are expressed at all time. And more so, it is only certain cells that express certain genes. For example, it would be silly to express your eye color in your fingernail. In addition, we have something that is known as housekeeping genes. These are running the day-to-day business in all cells for functions that all cells share. So genes have what we know as promoters, and there are a number of mechanisms that we use, and they all have their own effect on whether or not the gene is expressed at what rate and for how long. And some of these mechanisms take place in the nucleus, while some take place in the cytoplasm of a cell. Let's look at a difference of these. Well, ones that take place in the nucleus are shown here, and the first one of them, chromatin condensation, is a method that eukaryotes use to keep certain genes turned off. You can think of it as if the chromatin is wrapped together very tightly, very little if none of the genes are expressed. And we can see these with microscopy as the more tightly coiled up regions stain darker. We have already talked about DNA transcription earlier. There are different factors, activators, and so on involved, and this is a very well regulated process. So we have a lot of control over what genes are expressed when compared to simpler organisms. We notice also that not all the DNA leaves nucleus. What we do actually is rather clever, and this takes place during mRNA processing. So you can think of DNA as containing a lot of unnecessary information. Say if you want to cook a cake, you do not need to learn about the structure of eggs or how flour is made and so on. And all this unnecessary information is in the introns. So what we do is that we remove introns from this RNA and combine only exons together. And it is only these exons that get to leave the nucleus. Now I know that these terms may sound a little bit counterintuitive, but think of it this way. Introns have no value to making the actual product, and they must stay inside the nucleus. Exons are what we really need, and they get to exit the nucleus. So those were our control mechanisms inside the nucleus. Let's look at those outside the nucleus. And here we go. mRNA translation involves these special proteins in cytoplasm, and they regulate whether the translation will take place or not. And of course, this is where environment may play a significant role. For example, something is not made unless we have the right ingredients to make it available. Well, this next step, protein activity, refers to the fact that some proteins are not active immediately as soon as they are made. For example, your textbook mentions that insulin must be processed before it takes its active form. And this is useful because it allows us to delay the protein's activity until it is needed. And finally, as we finish off, I want to mention that of course cells of a multicellular organism signal between each other. So this can be to control what a certain stem cell becomes to, or control growth and daily functions of a more mature cell. And we can see many signaling pathways. And these can be quite complex, really. And these two that I'm showing here are not even the most complex ones that are out there. So this is where we will wrap up this talk. And we will look at the prokaryotic gene expression next. Good job with sticking around this far.